Hi, my name is Sara Jafri, and my project is about automated CT analysis using image processing. Now, head injuries are common, and a specific severe type of head injury is a traumatic brain injury, also known as TBI. TBI diagnosis in the earlier stages of the injury is very crucial. However, this is very challenging, as it is difficult to detect the microstructural changes in the brain. The only proper way to determine whether the patient has TBI or not is for a qualified physician or a team of qualified physicians to sit down with the CT scan images and meticulously examine them to see if they can find the TBI regions in order to properly diagnose the patient and begin the treatment. Now, this is not the best use of the physician's time that could be better spent treating patients. Also, the longer it takes to diagnose the TBI, the more the TBI develops, making it more difficult to treat. Therefore, it has become essential to develop an automated program that will allow physicians to simply input the CT scan image to receive an output of the location of the TBI region. Although there are methods that are capable of doing parts of the TBI region determining process, they are either semi-automated or do not consider the variation among different images. Therefore, as a part of my biomedical engineering-based project, I have been working towards developing an automated system, but only for a specific type of TBI, which is hematoma. In hematoma, there is a pooling of the blood towards the outer perimeter of the brain near the skull, such as in this image. Using image processing techniques, the way that we want this new system to work is that it would first adjust the contrast of the image to help make the different regions of the brain, such as the ventricles and the cerebral tissue, lighter or darker so that they are more distinct from each other. It would then recognize the part of the brain that was the skull and then crop that out, since we don't necessarily need the skull to find the hematoma region. Then, since there might be some shift of the brain due to the pooling of the blood, we register and realign the image so that the midline of the brain coincides with the middle of the image. Then what the program does is the part that is most important to us. It essentially uses a backwards type of facial recognition process in which it matches the image of this brain to what a normal quote-unquote brain is supposed to look like. And it looks for any discrepancies or differences between what a normal brain would look like and what the brain that we are dealing with looks like. One of the major discrepancies that we are looking for is the pooling of the blood, specifically the hematoma TBI region. The algorithm then highlights the possible hematoma region using image segmentation techniques and outlines the perimeter of the brain in red, as well as providing its coordinates, which would then allow the physicians to focus their attention to those regions and easily determine whether the patient can be diagnosed with TBI or not. As you can see, these results are fairly crude, and though they are not the desirable results we are looking for, we have been working towards refining them for better accuracy in the future, such as in this image. At this time, I would like to thank my friends and family for their support, as well as my research mentors, Dr. Najarian and Dr. Suresh Meher. I would also like to thank my current peer advisor, Patrick, and my former peer advisor, Sharmise, for all that they have done for me. I would like to also thank the University of Michigan Medical School for the brain images that they are allowing us to use for both our project and for this presentation. I would also like to thank UROP for allowing me the opportunity to be a part of this research project experience. Most importantly, I would like to thank you for watching.